that's what I'm working on. I work on Gelycioida, and I'll touch on that. So this is a brief statistic of the diversity again. So in the world, this is a 2011 census kind of statistics. Microlepidoptera, 57,911 species. Macro, 15,000. Total is 73,000. And out of that, about 18,000 butterflies. In North America, we have 830 butterflies out of that. This is mostly in tropical areas. About equal number, more or less, of macros and micros. And total of about 1,200 species. And that's just a described diversity. That's what has been named. I'm just going to show you some pretty micro-moths. That's the title of this picture I chose for the poster. And that's a Coleophora trifoli. You don't get this in the Yukon, but it comes, I think, all the way down to Alberta and southern BC and such. And it's a really shiny, pretty thing. I'll talk about coleophoras a lot more later. That's a species in the group that I'm working on. It's a Normashima, and it's a species that we described 3-4 years ago now. It feeds on Schafferdiae, and that's what we called it, Normashima Schafferdiae. It's from Quebec. It's a live specimen, lab reared, so, and that is about, I want to say 3-4 millimeters, or something like that. You can see the individual scales. So these guys are really small. So the scale bar, by the way, is about 2 millimeters. So that would be about a centimeter from tip to tip. But the wings are mostly narrow. In most of the Microlepidoptera, the wings are narrow, and the real estate for exhibiting pattern and color and showing off is very small, but you'll be surprised how creative these guys get. So there's all these different patterns that all follow the same kind of orange, silver, black thing. And there's 7 species from 7 different families here. So the top 3, I don't know if you can see these are Cosmop trigids, Cosmop, Elachistid, different family, Gelyciid, Heliodinid, more Heliodinids, Momphid, and Eucophorid. Different families, they all kind of look more or less alike. To a non-expert, he would probably put all of these in the same genus. That's one end of the spectrum. The other end of the spectrum is something like this. How many species do you think we have here? Somebody said 1. You're good. You're good. This is called Aclaris variana, and the name is variana, so it varies. And this is all one species, believe it or not. Some of these guys came out of the same egg clutch. So basically, morphology doesn't really work in these guys. And even though we think, you think you've collected enough to have seen everything, you still get new variations that nobody's seen before. So there's this and there's that. I just threw this in because, not only because it follows the same orange-silver pattern, but the name is fun to say. Decanticincta diticinctana. It's from Japan. Try saying that 3 times. It's a tortoisid. This moth was described by my friends Bernard Landry and Almos Solis from Smithsonian. It's from Galapagos. They named it Mordor. Desmia Mordor. It's kind of fitting. 9 species in 1 genus. This is artwork by Patricia Gentili-Poole from the Smithsonian again. This is genus Ativa. We have 1 species in southern Ontario, but then from there it goes all the way to southern South America. And these are just the forewing and hindwing kind of cut off and put them. I don't know if this is Photoshop or real. It might be real because they have so many specimens there. And then when you get to the tropics, it just goes crazy. This is a photo by a good friend of mine, Paul Bertner out of BC. This is from the Philippines. The Philippines, like the Yukon you have here. It's ants mimicking, supposedly mimicking ants or something with its legs. The moth keeps its legs up like that. maybe spiders. It's mimicking something, we just don't really know. It's so striking. We couldn't figure out what this was. The micros in the tropics are so poorly worked out, and it took a lot of work for us to actually find what it was through a colleague in the British Museum who dug it up, and he said, it's probably this thing here we have from 1800. And it turned out to be that. This is another species in the same genus. Again, Paul Bertner photo from the Philippines. Completely different wing pattern, same genus. We don't know what species it is. It's probably undescribed. This is a gracilarid Caloptilia. You get this in BC. It's just a funny sitting pose. Most of the species in Caloptilia have this sitting pose. It's like they're sitting on a throne or chair or something, but they're really tiny, as you can probably see from the leaf. Gracilarids are mostly leaf miners. This is another gracilarid from Europe, a Phalaenorhector, and that's what they do. They mine the leaves. So that's one type of life history that you see. I'll talk more about what other things these guys do. They have a wide diversity of biologies. This is another small moth out of Colorado. It's a heliodinid. This is my photo, and that's 1 millimeter. So this is, again, less than a centimeter from a wingspan from tip to tip. And it's just fascinating. When you look at these things under the scope, it's just those wow moments. More interesting groups, plume moths and mini plume moths. These guys are really showy, show off, and kind of neat looking. I have a couple of books here that you can feel free to look after the talk, just to, if you want to see more micro-moths. There's so many that I just didn't know what to show. But like I said, not all of them are small. So you get things like that. It's a cosset from, I believe, Australia. And that one is a Hepialid, different family. And that one is a Casnead from South America. So 3 different moths, 3 different families, all considered micros, but they're huge. You have things like this. This is a bagworm, the Lica, and the females are completely wingless and flightless. And that's the male. And that's what they make, and they pupate inside a bag of sand and such. And they're algae feeders and things. So you don't know the sex until the adult comes out, and if it's a male, it flies away. If it's a female, it stays there and just crawls around, just drags this thing with herself. This is the only moth that I know. It was described in 1912 by a German entomologist as a diptera. So this is the only moth that I know that it was described originally in the wrong order. So this was known as a dipteran for a long time, because what the heck is that? Psychidae is the family, yeah. Probably it comes from Psyche, the Greek myth of Psyche and Cupid. It's a mythological story about, I don't know if you know the story, but it has to do with, Psyche is generally the Greek and Latin word for butterflies. Right, and then some of the more, like the 3 I showed you in the first slide, that one of the first branches is Micro-moths. This is really close to caddisflies, but it's a moth. And 3 of these early branches, they don't have the coiled proboscis as most moths and butterflies do. Their mouth parts are different. They have chewing and biting mouth parts, so they're really strange. But that's what they do. They do look like caddisflies, but they're not caddisflies. This is out of Croatia. That's something I got through Destiny. So diverse biology, they do all kinds of things. Microlepidoptera, as I told you, it's a huge umbrella covering a lot of things, and you can find gall makers, leaf miners, stem borers, seed borers, fruit borers, fungus feeders, detritivores, scavengers, even parasitoids, and so on. Some species of Pyralids, Acanthropinae, one subfamily of Crambidae actually have aquatic life cycle. So like mosquitoes, they're completely dependent on aquatic habitats. They feed on almost anything, from liverworts all the way up to angiosperms. You can find them almost anywhere, as long as there's enough temperature for them to be active. And they are very important elements of biodiversity. That's a gull, by the way, of an Ormashima on a, I think it's a goldenrod. That's the group that I'm working on, so that's one of the gall maker moths. A quick pie chart of the North American species. So if you remember, again, the trees, some of the branches were thin, some were thicker. In North America, we have like 7 or 8 groups that are more species reach, like Totricids and Galichiids, Gramids, Pralids. The rest are mostly smaller families. And the take-home message here is that they're very poorly studied. Like from barcoding work, and I'll talk about this after this, we know that there are 275 coleophores described, but we actually, right now, we probably are guessing it's about 650, because we have more barcode clusters, unique barcode types, than 275. Same with Galichiids, same with Schitrids, for example. So 60% is undescribed, 45% is described. This is probably 90% undescribed. So we need people to actually study these and describe them. I'm going to switch to barcoding now. So this was micros. So this gets into a molecular side of the story. So if you remember your high school biology, you probably remember that all these organelles in the cell and kind of what they do. For DNA, all you need to know or remember is the nucleus, which is where the genomic DNA is, the nuclear DNA is, and mitochondria, which is the powerhouse of the cell. And these two are the only ones that have DNA. The rest of this, they don't contain any DNA. The nuclear DNA is important because it's what defines the organism. That's in the process of meiosis, that's what, half of it comes from the one parent and half of it comes from the other parent, and it's very critical in the next generation, in the chromosomes and so on. And there are lots of control mechanisms over it. And so that is different from mitochondrial DNA, which is a DNA only for the mitochondria. And it's not in the shape of chromosomes, the mitochondrial DNA. There's little circular pieces of DNA, and there's hundreds of copies of them in each mitochondria. And these circles are small genomes, mitochondrial genomes, containing a few genes that code for things that are used in energy production for the mitochondria and for the cell, and respiration. So imagine this as a strand of DNA, like a round, kind of elastic band strand of DNA, and with different parts, different genes, and different colors. Part of this is a complex of 3 genes here that are called cytochrome oxidase 1 and 2 and 3. So these 3 genes are a complex of genes that have a role in respiration. I don't want to get too technical with this. One thing to see here is 5' pointing this way and 3' pointing that way. And it's like that with every gene. And I'll get to it in a minute. The DNA barcode region is somewhere there. So this piece, cytochrome oxidase 1, is about 1600 base pairs. And I'll tell you what base pair is if you don't know. And the barcode region is just a portion of that, about 658 base pair of it, closer to the 5' end of that gene. So that's the unrolled strand of DNA. Imagine that was the circle, and then if you unroll it and flatten it up, this is what you get. So this is what a base pair is, and you get your 4 nucleic acids and your adenine and thymine and cytosine and guanine, and then that's how they pair up. And that's how they're transcribed. So from 5' to 3', you have A, T, C, G, blah, 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 and then these are kind of grouped with the opposite on the other end. So this is called a reverse complement strand. But the convention to read DNA sequence is from 5' to 3'. So when you see a sequence like this, it's the read from the 5' end to the 3' end. And then if you reverse complement this, it'll be a completely different sequence, but essentially the same, read the other way around. So that's what a DNA barcode looks like. So for each of these 4 letters, A, C, T, and G, there's one color, and then when you translate that into that color, that's what you get. This is straight off the BOLD website, which is what I'm going to get into in a minute. So what do you do to get this? It starts with field work. So the first step is actually going to the field and collecting and or putting a sheet up and collect all the moths that come to the light, and then pin them and spread them. And this is what we use for micros. This is called a spreading board. It's just a small box that big with grooves cut in styrofoam, and then you just pin them in the field and prop up the wings so that you can see the pattern. And then they get labeled and double mounted in the lab. And so everything is vouchered. All the DNA samples get a blue label saying the legs were taken off of them, and they're stored. This is the national collection. And so for DNA barcoding, what we do is that we take every specimen, take a leg off of it under the microscope. Accidentally, this is from white horse. And each leg goes into one of these little wells in a lysis plate. There are 96 samples, 95 samples plus a control here on each plate. And they're sent to the Center for Biodiversity Genomics in Guelph. That's the actual building. And so what they do with these plates is the first step is to lyse them. It's called a lysis step, the first step. And they go on these robots, and they can process up to, I don't know, 100 plates a day or something. And so the first step is to dissolve the cell wall and to release the genomic and nucleic and mitochondrial DNA. Both of these together are called the genomic DNA. So once the cell wall is dissolved, lysed, and the DNA is released, and it can be targeted, and the barcoding region can be amplified, and for that they go on these PCR machines, polymerase chain reaction machines. And what they do is that they put things in there, think of them as magnets that find the barcode region in the genome, and they start copying that region, hundreds and hundreds and thousands, millions of copies in these machines. So in the tube, when you had one copy, then after a cycle that takes about maybe 40 minutes or so, how much time do I have? 22 minutes, okay. You get millions of copies of that. And then after that, they're sequenced on these machines, which are called sequencing machines, different machines. And the sequences are read with laser, and this is what you get afterwards. So the results come in the colorful bands, and there's a software that reads them. And this is actually the Guelph lab, so you see the PCR line up there. So from here, you get these nice chromatograms, and this is what the laser gives you. So the laser goes through these reads, and it translates these reads into alphabet, like the 4 letters. So for each of your samples, you get the DNA barcode region, and you can immediately on the machine, on the computer, you can see where the differences are between your samples. Like here's you have an A, and on the other sequence you have a T, so that's the difference. All of this data is uploaded into the Barcode of Life database, data systems, and it's a website. Most of the data is public, but there's also non-public data. So if you're collaborating with people who want to publish a paper and they have data that is not released, they can make it available to you. Otherwise, you can just go and explore what's public. There's a lot of public data. So this is what a project page looks like on the website. This is a project on micro-moths of North America. It's how many samples, who's got access to them, where they are deposited, what institutions they come from, what are the age of the samples, the sequences, and so on. Each sample gets a page, like a specimen page, which highlights or details the collection, specimen details, what the taxonomy is, where it was collected. There's a map, and whether the sequence is public or not, or the record is public or not. Again, each specimen gets a sequence page, a different page, with the barcode information. So there's a specimen page and there's a molecular page. So on this page, you can see, you can actually, after processing, this is where the barcode goes, and all the chromatograms and everything becomes publicly available immediately. And there's a link here. So on this page here, it says gene bank accession. So as soon as these are uploaded, they automatically get a gene bank number 2, and they go on the gene bank database. And if you click on that blue link here, it takes you here. And then this is so the two databases talk to each other. That's the beauty of it. So once your data is up, once your samples are sequenced, you can go to the project page. Here are all the tools that you have. This page, yeah. So here are some of the tools that the website gives you: taxon ID tree, distance summary, sequence composition, and so on. So you can play around with this, and then what most people do right away is to generate trees like this. And this is a neighbor joining tree, which gives you the distances between your samples. So say you want to generate a tree on genus Arotrura, so you go there and search Arotrura, and you get 100 or whatever sequences, and then you generate this tree. What this tells you, like the horizontal lines, that's 2%. So say between this bunch, which is all the same species, there's essentially very little difference to no difference between your samples. It's less than a fraction of a percent here between those. So that's all one species. Between this one and the next sample, there is about 2% here and about 1.5% here. So that's 3.5% between these two. And normally in Lepidoptera anyway, between two well-recognized species, you have at least 2% divergence. So that's kind of a yardstick. If you get a 2% divergence between your samples, you're probably looking at two different species. If you get less than that or a little more than that, then you have to kind of be careful. You have to examine some other characters and see what you're dealing with here. And the longer the branches, obviously, the more distant your samples are. So these would be all the same species, probably. A little bit of variation going on. So if you want to investigate that, maybe they're from different places in Arizona. Maybe they're different subspecies or something. Maybe not. But whatever these are definitely different, more than 2%, more than 4% from whatever comes next. So the vertical lines don't mean anything, and the relationships here at the deeper nodes really don't mean anything. So the neighbor joining is a very simple distance analysis. It's not a phylogeny in the sense that it's not giving you the evolutionary history of the group at all. It just tells you between what you have here and what you have here, there's 4% difference in the barcode. That's all it tells you. But it's presenting it to you in the form of a tree, and that's what misleads a lot of people. They look at this as an evolutionary tree, which is really not. It's the simplest form of genetic analysis, really, distance analysis. So once you have this tree, you can also have a snapshot of all the photos for every record in that tree, and you can just eyeball them and see if they're different. So as I mentioned, 2% is kind of the yardstick, and so within the species, these are Micro-Lepidoptera families in North America. So within species in each of those families, so we're looking at one species, different samples from the same species. This is the kind of variation you see. So it's under 2% most of the time. And when you look at two different species or different species within one genus, so say we have a genus with 5 species, the average distance between those species normally are those red ones, red squares. And the triangles are between genera in each family. So say you have different genera, and then the average, it goes up. So the distances go up normally. It varies a lot. So yes, it's a yardstick, but again, you get cases where this gets very low, or sometimes it's messed up, and then you have to see what's wrong. Is it the taxonomy that is wrong? Is it something wrong with your sequences? I'm going to give you some examples of what barcoding does for us using this group, which is Coleophora. Again, the title of the talk, the photo from the talk. And these are case bearers. So in their life history, again, something a different kind is that they make these cases which have different shapes, and they're called pistol cases and cigar cases and some silly names like that. But the larvae lives in those cases, and it kind of drags it along with itself until it pupates inside of it, and then the adult hatches and flies away. So sometimes you find cases that are empty and the adult is not in there. That's what the adults look like. So they're, yes, some of them can be really shiny, like the one I chose for the poster, Trifoli here. But for the most part, they're drab and they're very similar. So sometimes you can tell, yeah, you have a group of specimens that are whitish, so maybe they're the same species, but when you get hundreds and hundreds of them, it becomes impossible unless you dissect them, like every single one of them. And dissection, that's a male genitalia, that's the phallus, that's a female genitalia. It gives you a lot more characters, and it's very useful, so it's essential in Microlepidoptera taxonomy. But it's a skill. It's a very difficult skill to learn. It's time-consuming. It's expensive. You need to have the tools. You need to have, the right microscope and everything. And there is actually a paper from 1976 where somebody said Lepidoptera shouldn't do more than 6 genitalia dissections a day because, your eyes get confused and you may screw up. It's really funny. So what do you do? Can barcoding really help with this problem? That was the first approach that got us involved in barcoding back in 2003, where when I went to Guelph, barcoding had just started, and the collaboration between the Canadian National Collection and the Guelph team had just started too. So that was the first main thing. Like, if we can do something to minimize the need for dissection, that would be awesome. So it turns out that yes, barcoding can help with that, but not only that, a lot of other things. So barcoding can help with sorting species, detection, discovery. It can detect new cryptic species, undescribed species. It can help in diagnosing problematic species, species that have difficult taxonomy, if you barcode the types and compare them with other specimens. Another problem is association of males and females. When you collect a long series, and I'll show you an example, you don't know which male goes with which female sometimes. It can show you some flaws in the taxonomy. Sometimes you have synonymies that you're not sure, and if the sequences are the same, that's a good indicator. Identification of otherwise unidentifiable specimens, taxonomic placement of problematic species, so on. I'm already half an hour in, so I'm going to rush through the rest of this. Example of discovery of species. These were all collected same night, same event. They all look the same. Barcode them, 4 different clusters, 4 different species. Dissect one of them, figure out what they are. You don't need to dissect the rest of all of these. Dissect one of them, you know that's that. This is a new species. You dissect it, genitalia doesn't match anything, new species. So Coleophora, the black lines are the described species. The red lines are all the ones that we've found that have no names, new undescribed species. So this is what happens usually when you print these trees and go through them and you figure out, so what's going on here? I have Bactra, Virtuana here, but then again, I identified those as Virtuana. Which one's correct? Then you dissect this one, and one of them, and then you figure out, this is wrong. Change that taxonomy. Here, you don't have a dissection, and none of these are dissected, so dissect a male and a female here. Dissect one here. So instead of dissecting 100, you dissect 4 to figure out what species you're dealing with. Example of something we taught. So these are after you dissect. This is the male genitalia, and then we found that this thing we identified as something that was here. The genitalia was very similar, but it was actually a new undescribed species because proportionally there were differences in some minor things that turned out to be a new species. Our identification was incorrect. With NGS technology now and some other methods, we can barcode old types now. So this is very helpful in clarifying ambiguities with identification. So these were all the types that we have barcoded so far, like from 1871, something we barcoded. And then you get, for example, we had all these specimens and the barcode kind of nicely matched, fell into that group, which was, yes, our identification was good. Sometimes you have a specimen and the holotype matches it. And sometimes the holotype is a male and what you have is a female and you were not able to identify it before. And now you know that it's that species, the female of that species. And then you can associate sexes too here. Like, again, all specimens from the single collecting event. Then you get all these clusters, different species. Some are males, some are females, which was otherwise not very easy to associate. It's a direct quote from Ron Hodges, who wrote this book in 1976, and he says, I'm describing 4 new species here that are known only from females. Probably somebody in future will figure out that these names are not necessary, and they can synonymize it later. So what do you do? So this is before DNA. Now we don't have that problem anymore. If we can sequence them, we know what they are right away. Again, another example. A female from Ontario, it was reared, and the 3 males from Indiana, they matched 100%. We know that it's sternuata now, what we have in Indiana. Detection of possible synonymies. These 2 were 2 different species, albovanescens and what's the other one? Tilifoliella. Tilifoliella is darker, albovanescens is white. And there was very little difference between them. And then we looked at the specimens and we found that it's actually a gradient, that you get all the colors from dark to the white, and it's all one species. So they're synonymized now. This is a paper I did with a botanist friend of mine who reared this thing, who collected this thing from an endangered aster in St. Lawrence Island. And we couldn't rear them, they failed, so we didn't know what it was. We knew it was a coleophora because it had a case, but we didn't know what coleophora. And the host, there was nothing recorded on that plant, so we barcoded it. We barcoded the larvae, and it matched some adults that we had barcoded of Triplicis. So we were able to put a name on this thing that was otherwise unidentifiable. Taxonomically, this was discovered in Quebec in 2006. This is not a micro, by the way, it's an actuid. The author, Louis Handfield, couldn't place this in a genus. He didn't know what it was. He was sure it's a new species, but what genus it belongs to. Barcode placed it close to this genus, Capis. So he described it under Capis as Capis archaea. Helped him that way. This is also not a micro. Another paper I did with Chris Schmidt and others. This is an extinct moth from Jamaica, the Jamaican sunset moth, last seen in 1895. We had 1 specimen in a collection we barcoded, and we were able to figure out its phylogeny, basically. So now we know where it fits on the evolutionary tree. So barcoding also helps us figure out introductions and so on. So this was a species known from Europe that we found first from BC. This is Denmark, and this is Netherlands. So we had 2 specimens that came out like identical to the European specimens. And since it's Vancouver, it's essentially a big red flag of introduction right away. Sometimes the introduction happens the other way around. Sometimes it's an American species that is introduced to Europe. Blastobacillus glandulella was found in northern Italy, I think, first, and then from there it's now spreading elsewhere. It's funny, Europeans found this thing in Italy and they didn't know what it was. They thought it's a new species, so they named it. In 1994, they put a name on it, not realizing that it's a new world species. And then we figured it out before barcoding, actually. We saw the papers, like, oh, this looks familiar. And then we asked them to send us their specimens, and we barcoded it, and sure enough, it was that. So, and there's a American Navy base right next to where this thing was found first. So it's Americans who did that. So that's how it went. Another interesting case is some things that you just have to scratch your head, like this thing found in Carcross. It's one of the species that I worked on. The closest match to it is from the Ural Mountains in Asia. Nothing is known in between. It's probably an artifact of poor collecting, like nobody has really sampled this thing. It's probably a Beringian species that goes over here, and it's probably widespread, but right now, the 2 dots that we have on, or the 3 dots that we have on the map are so disjunct that it's just bizarre. I actually borrowed, I found this among the specimens I had from the Carcross, and I didn't know what it was until I looked broadly at the literature, and I saw the genitalia drawing in a Russian paper. I'm like, this is what we have, but I wasn't sure. So I borrowed the types, and we barcoded the type. So that's the type from Russia, and that's our specimen from Yukon. There is a little bit of divergence, but it's the same species. With this, I'll just briefly touch on the project that I did for the Carcross Dunes. So this was part of my PhD thesis, actually, and because of their importance in conservation priority, Environment Canada actually was interested to know what was there on the dunes, and there was a broader work that tried to identify all the Lepidoptera before I came in, and they also looked at some of the Normoshamines, and there was no barcoding done then, so they had sorted things out morphologically, and I knew that with this group, morphology doesn't really work. So again, the 4 moths that you see up there, they're all the same species. So initially they were identified as 2 or 3 different species, but barcoding showed that they're identical. It was just male and female or some variants that they're we barcoded about 350 specimens, and we got this nice tree, which gave us about 21 species, and 9 of these are still unnamed, which doesn't necessarily mean that they're new. It's just that we haven't been able to find what they are. And so my results was different from what was there before. And the sections also helped very nicely. What else do I have here to say? So that's what the genitalia looks like of males, and those are the females of some of these Yukon Carcross specimens. The male genitalia structurally, it's like a cylinder. It's like a round thing, and the phallus is inside. And because it's so 3-dimensional, you can't really see the characters very well unless you kind of unroll it open. And this is what it looks like. If you cut one side and kind of unroll it and flatten it, then all of a sudden you can see a lot of characters that were otherwise invisible. So that's the method I used, and for the females, this is what they used to do previously. They just left the abdomen attached. Now I pull the whole thing out, mount them separately so I can see everything very clearly. And that's the kind of variation you see in closely related species. There are minor things, minor size differences, and minor placement differences, and so on. So that's what most of taxonomy is. You have to rely on what you see under the microscope on the genitalia to define and describe your species. I haven't been able to conclusively say that there is an endemic species to Yukon that is found nowhere else. I just don't know that yet. And we need to collect a lot more, especially in Yukon. It's so undercollected. We have very few dots, mostly on the Dempster Highway, actually. So yeah, so that work is still ongoing, and this result I just got last week, and I'm still working on it. So there's this thing from Rampart House, and I told Sid about this. It's a completely insignificant moth. It's just a little brown thing. But when I dissected it, this is collected in 1961 or 51 by the survey people who went up there. And I dissected this, and I was like, well, holy, what the hell is this thing? And I couldn't even place it to a genus at the time. So through networking, we figured out that it's Pogoketia, which is a new genus for North America. And it's only known from the Mediterranean region. So, but also, there are a couple of related species in Central Asia and Iran, southern Russia and Iran. So although it's a new species, for sure, it's different from what's there, but it belongs to the genus, which is a new genus for North America. And now the barcode that we have confirms the genitalia identification, which is really neat. So I have to work on that. All right, coming to an end here, which is good timing. If you want to get involved in Lepidoptera, what do you need to do? This was a thought that came late to me, actually. When I arrived here, I said, maybe I should probably guide people who want to get involved in collecting micros and figuring out what's here. But then I thought, well, it's a bit complicated because you need to, there's a bit of training involved. So you need to know, well, you can photograph them, but if you send me a photograph of a moth like the Variana thing, what I'm going to say is, I don't know. I need to see the specimen. I need to dissect it. I need to barcode it. With micros, photos normally don't work. Sometimes they might. Most often they don't. But yeah, try to participate in bioblitzes. Try to see those micros. Try to find them. Figure out how they live. If you see artifacts of their presence, like the leaf mines or galls or things like that, you can collect them, keep them in plastic bags, and keep them in your house and check them out sometime regularly and see if something comes out of it. Sometimes you'll see moths flying in your bag, which is really neat. But you need to have some tools. You need to have vials. You need to have spreading boards. You need to actually see somebody pin and mount and spread a micro to learn how it's done. So if you're really interested in doing that, it's not really complicated. It's just that you need to see how it's done, and I can show you, but it's an involved thing. Get in touch with Sid. Email me. I can show you how to do it. I can send you some material, and you can start from there. And that's my coordinates. That's my email, and you can find me on Facebook and Instagram and Twitter and whatnot. If you have any questions, I'll be happy to answer. Yeah. Thank you. I have a 10-minute video that I can show you if you're up to it. It's about a micro-lepidopteran from Hawaii that a friend of mine made. But we'll decide if you're interested to see. It's 10 minutes. Yeah? Okay, let's do it then. There are not a lot of videos on micro-lepidoptera, so this was really new to me. I actually saw it in the Entomological Society meeting in Florida. So it was premiered in the Entomological Society. It's professionally made, so it's really neat. So here we go.
Insects in Hawaii, they make up a huge percentage of the fauna. they're really specialized and particular to where they can live, what they can eat. It's important that we learn how to protect them.
So Philodoria micro-moths are the leaf miner that lives inside of the leaf. They're only found in Hawaii. These are caterpillars that burrow into the leaf's layers and grows within those layers. So it's protected by the leaf. The caterpillar will keep eating, and eventually the mine oftentimes becomes really big, into a big blotch. When it's full, it comes out of the leaf, cuts a circle. The caterpillar goes into this disc and wraps itself from both sides. Then, in several weeks after that, the cocoon will turn into a moth. The moth will kind of push itself out, and then it flies away. I was curious to know if these moths still existed since the works that were done approximately 100 years ago. And to my surprise, we discovered a few, and we're trying to reassess the changes that might have taken place within the last 100 years, whether some species have gone extinct, and so forth.
So can you introduce yourself, give your name, title, and affiliation?
So my name is Akito Kawahara, and I'm a researcher at the Florida Museum of Natural History. My official title is Assistant Curator, and I've been here for 4.5 years now. My favorite insect, it's a great question. I think it is a moth. I really like Philodoria. I think it's one of the really cool groups. They're pretty unusual in terms of their distribution. They're only found in Hawaii. We don't know how they got there, and once you look at them through a microscope, they are truly spectacular. This one is pinned because it was collected by a researcher many years ago. It's preserved as a museum specimen and has historical data records on it. Yeah, these moths are really small. Some of them are 10 millimeters at most in width. Some of them are only 2 or 3 millimeters. The patterns on the wings are actually quite different, and this is one of the ways we can tell that they're different species.
They're all little tiny guys. They're all done. Oh no, there's one right there.
So one of the very unique aspects about this group of moths is that the plants that they feed on are very unusual plants that are only found in Hawaii. So they're endemic, and also many of them are threatened. So the scientific goal in studying Philodoria is to understand the current distribution, the evolution, and also the conservation of this really unusual group of moths that's only found in Hawaii.
Asperomania. Okay. My name is Kiahi Bastamente, and I'm from the island of Maui, Hawaii. I like to think I work for the Hawaiian people and the plants and animals native to here. This is Sonicula sanosensis. It's got a tiny flower. So Hesperomania is an endemic genus to Hawaii, and it's in the sunflower family, and it has this thistle-like flower that has tons of nectar in it when it's fresh. Flowers maybe like that big. Beautiful flower. It's become pretty rare. We think it's lost its pollinator. It has a hard time moving pollen, even though it still does. It's almost doomed to extinction at this point.
There's maybe 100 plants known on Maui. The split stigma.
Oh, yeah.
We found out through research in the herbarium that there was a Philadoria associated with Hesperomania. I think that was for Lanai. Hesperomania is probably gone from that island now, but figured gotta be on the other islands, and so started trying to look, and sure enough, there it was. Made a couple trips to make some collections to see if it was for sure, and looks like it is a new species, one for each island. There might need to be a certain amount of Hesperomania around in the population in that local area for them to survive. Knowing that, and the amount of plants that are still remaining, the Philadoria may be more threatened than the plant.
There's a lot happening in Hawaii in terms of habitat destruction. Many people, when they visit Hawaii, they see green mountains and it appears very beautiful. But in reality, what's happening in Hawaii is quite striking. One of the things that we want to do as human beings is to make sure we preserve what we have around us. Human disturbance can have a potential effect on the plants and thereby affecting the future of these insects.
As far as the culture goes, our culture depends on the existence of these moths and other insects and plants. Our culture was founded on these great animals and plants. It's talked about and put into songs and legends. It's documented that we respect these things and they respect us in a way that allows us to survive. Like any people, they're dependent on their resources. I don't think there would have been a possibility of us succeeding as we did without all of this. I don't think we'll be able to move into the future without it.
I believe there's possibilities of more Philadoria species to be discovered. These last couple of years, I think, have been a really good first step to assess the current status of these moths. We also don't know what's going on the northwest Hawaiian Islands, if there are any Philadoria left. So there's certainly places that we have not looked that could potentially lead to more species.
Okay. Feel free to have a look at the books here and here if you have any questions. Yeah, thank you. Micro-moths are not the only thing that mine the leaves. So it could be a beetle, it could be flies. An aspen, yeah. Those they're most likely the Philocnistis. Yeah, it's the same family as these. Yeah, the white little things. They are. Yeah. Popular. It's common. It's not restricted to Yukon. I've got them in BC. I've got them in Alberta. Bruce. Same question. Yeah. Right. I just don't know, honestly. Gracillarides are not my forte, but they're, from what I know, very few micros overwinter as adults. Maybe those guys do, I just don't know. But I know that they're very difficult to see, first of all, unless they come into your face in flocks. That's how you notice them in here. Yeah, so unless you have an explosion like that, you don't normally see them because they're so small and they're very difficult to pin and spread too. So. Right. As a bonnet. Yeah. Sure. Go ahead. That was for North America. Yeah. That's a very good question. I don't have the numbers right now, but our checklist just came out a couple of weeks ago. A checklist on the Lepidoptera of Canada and Alaska, and I think there are some numbers that they. For the Yukon. Okay, there you go. Probably. The thing about Europe and North America is that our stuff is so poorly studied. We just don't know nearly as much as they do. They've been on this for 250 to 300 years. And I can count on both hands how many Microlepidopters there have been in the history of North America. So there are so few people studying these things, and we just know close to nothing about them. So. You get the sense that they're pollinators, but they're not really, no. They just feed on the plant. They thought that whatever was pollinating, there was no mention of an insect. They said it could have been a bird, for all I know. Right. Just because Philodora feeds on this plant, which is also endangered, so they're both kind of. If the plant is doomed, the moth is doomed too. So. Right. DNA degrades over time if it's not kept in ideal, like good conditions, like in the freezer or something. If it's kept in the room temperature, it degrades over time. That's why museum specimens normally, if they're over, I want to say 10 to 20 years old, they don't, the regular processing of DNA doesn't, DNA barcoding doesn't work. So you have to go through a special processing, which is a little more expensive and it's more involved. So what happens is that DNA breaks into smaller pieces. So you don't have that nice 658 base pair strand anymore. You have that piece is broken into chunks. So you have to target smaller pieces and then try to amplify them, and then you get those smaller pieces assembled together to get the whole thing. For the current new genomic methods, what people usually do is that they put stuff in alcohol right away as they collect it, like just put it in 95%, or freeze them in liquid nitrogen if you can. It's not too bad actually. I think if even for micros anyway, it seems to work even if they're pinned and they're less than a year old. You can still get very good quality DNA for genomic work. But it's kind of a hit and miss. You really never know. Sometimes the fresh specimens don't work. Sometimes old specimen you get good results out of. So I mean, it's not a clear-cut law that even after this many years, we can't get any DNA out of it. We get DNA out of again, it's a different technology. But like you saw, we get DNA out of stuff that is 200 years old. But it's a completely different approach to sequencing. Harm to DNA? Not really, no. No, it's actually recommended to keep stuff in -80 degrees freezers if you want them to remain intact for a long time. So the colder it is, the better. But if they go through freeze and thaw cycles multiple times, then that kind of damages the DNA. They're not on the wings. They're on the fringe. They're on the edges of the wings, and they're not really hairs. They're kind of modified scales that kind of pop out like that. They have some function in flight. So because these guys are so tiny, and if they fly, I think they have some function in flight. I'm not really sure what, but they do have some modified scales on the wings as well. Some raised scales sometimes that emit pheromones. They're used in mating and so on. They're called anthraconial scales. So there's that. We know a little bit. I mean, we know their life history, so we know that's what they feed on, and sometimes if their population goes crazy, they can actually cause real damage to what they feed on. We know they're food for a lot of things, like bats and birds, and you name it. So they have an important role in that chain, in the food chain. They are pollinators, like you saw with. Well, they can be pollinators. Some of them are for sure. Not the Philodora, not that one particularly. And what else? So they do have some roles. I'm not an ecologist, honestly. All I care about really is figuring out what we have. But yeah, of course, they're part of the ecosystem. So if they disappear, if they were to disappear all of a sudden, then we would know too. Do they hybridize? They do hybridize. No, I've read papers on this, but something I didn't really touch upon is that mitochondrial DNA is inherited through, it's a maternal lineage, so it only shows you the maternal lineage, and it's very difficult to detect hybridization through DNA barcodes. For that, if you're investigating hybridization specifically, then you have to look in some other markers in the genome to actually see that. But like I said, it's especially in lab rearings. I've seen papers where people talk about hybridizing some of the species. I don't think that's something that happens very regularly in nature. So, yeah. if I understood your question correctly, so if you're seeing the divergences that are less than 2% or more than 2%? Yes. Well, what you can infer if you're seeing, if you have something that you thought was one species, but you barcode them and they turn out to be different, then you can backtrack it to, you can investigate to see what they fed on, or you can investigate some other characters, genitalia and so on, and then you can find differences that were otherwise invisible. So the barcodes give you the first clue that what you thought was one thing is actually 2 or 3 different things. Or the other way around, what you thought were 2 different things are actually the same. So they might have different wing pattern, but the DNA says they're the same, so they might be variants of the same thing. Or they might be male and female of the same species that is sexually dimorphic. With pheromones and stuff, yeah, no, they're not relevant really. Maybe I misunderstood your question. Well, it comes from the moth itself, but the mitochondrial DNA only is good for telling species apart. Basically, that's what we do. If you want to investigate like deeper relationships, like how these things evolve, what the common ancestor was between different groups, usually you go with nuclear DNA, you sequence more of the genome, and you assemble a good data set, and then you start doing some solid, real phylogenetic analysis on them. You do likelihood, you do Bayesian analysis, and it's character analysis. So it's a different kind of approach than just, number, distance. Why it was chosen? Tradition, basically. So barcoding was, when it started, it wasn't new. Like people were already sequencing and then looking at different parts of the genome. And what seemed to be most working for most groups, I would say, in terms of determining the species and setting them apart, seemed to be the mitochondrial DNA. And the cytochrome oxidase 1 gene was very popular at the time. This was like 90s and early 2000s. And it had enough variation in it to be useful, and it was very easy to amplify. That was the key. Like it was, even if you had like older specimens, it was just very simple, and you always would get results. With some other genes, it's very difficult. Like if you go to the nuclear genes, it's very tough to get sequences actually, because there are very few copies of, unlike the mitochondrial DNA, as I showed, there are multiple copies in each mitochondria. So when you dissolve the cell wall, you get hundreds and hundreds of copies of mitochondrial DNA, while you get only 1 or 2 copies, very few copies anyway, of the nuclear genome. So ease of amplification, and the fact that it was very character rich was key to picking that particular site on one gene, on one mitochondrial gene. But is it especially different than any other region? I don't think so. It's just accident. Thank you.